So the next presentation is by Dr. Naga Malleshwari ma'am and Usha Suganya on the topic of plant nutrition deficiency using deep learning. Over to you.
Good afternoon, one and all. So my name is Naga Malleshwari and my colleague is Usha Suganya. We are from Networking and Communication Department. So our topic is about plant nutrient deficiency using deep learning. So our agenda like this, introduction, motivation, and then survey, and why we are choosing for deep learning. And later on from the survey, what we are going to find, what we found, that is findings, and finally challenges and gaps identified and references. So as we all know, India is a, 3/4 of the Indian economy is from agriculture, and many people living based on the agriculture. So we need to concentrate on the quantity and quality of crop. So as the technology is growing, we need the technology should help in improving the performance. Improving this quantity and quality of the crop production. If you do the same thing manually, it will be time consuming and error prone, and we also need the special skilled workers to identify the plant stress. So that's why we need automatic methods so that we can detect the plant stress early, so that we can take the preventive and corrective measures. So what are the advantages of automatic detection methods is we are going to identify the leaf, that is infected leaf, that is leaf stress, in early stages, and we also identify the types, the classification of stresses. So why we need to do the in early stage means so that the farmer can take the preventive and corrective measures to improve the quality of the crop. So if you see the classification of plant stress, especially the leaf stress, you have two types, abiotic and biotic. So abiotic stress is caused by the non-living agents such as ecological circumstances, weather changes, chemical use, and nutrient deficiency. And biotic is by the living organisms such as pathogens like bacteria, fungi, pests, and viruses. So this abiotic stress is non-infectious, non-transmissible, less dangerous. But even though they are not non-infectious, we cannot neglect this abiotic stress, because once the plant leaf got this abiotic stress, it will be vulnerable to the pathogens, so that it may lead to biotic stress. So, and this biotic stress is infectious and it will spread to entire crop. So there is a necessity of early detection of plant stress, especially the abiotic stress. So why we have taken this topic is, if we see the papers on biotic and abiotic stress, most of the papers are concentrating on biotic stress. Only 4% of the papers are based on abiotic stress, especially the nutrient deficiency. So we have seen in some papers, based on the survey, we presented this. So this is the nutrient name, and if that is become deficiency, the color of the leaf will be changed. These are the symptoms. That is, when a mineral is got deficiency occur for a leaf, it can be identified in the leaf by seeing the color. Okay, color and color plays a major role in identifying the stress deficiency. So boron, calcium, the slight yellow may occur at the tips of the leaves, and it turn brown to black and die off. That means later on the leaves will get die off. So this is for copper, and the images also we have pasted here. So finally, this is our findings. So this is nitrogen, phosphorus, potassium and all. The deficiency occurring means this is the symptoms. That is, what is the color change, what affects the leaves. So why we have chosen the deep learning for identifying the deficiency of plant nutrients means, so nowadays deep learning plays a major role in many agriculture applications, and even the plant leaf stress too. So, and we also have many pre-trained models, that is CNN models, and we have availability of data sets also plenty, and the cloud infrastructure is growing faster. We also have the GPUs, we can process the data or images using the cloud environment. So this is from our survey. We have seen that some of the models has been used in plant leaf stress identification. Here, plant leaf stress identification in the sense both biotic as well as abiotic. Abiotic stress papers are very less. So from our survey, we found that leaf of the color plays a major role in identifying the plant leaf stress at earlier stages. As of now, up to our survey, VGC 16 and VGG 19 is giving the better performance. So the later on slides will be discussed by my colleague.
Thank you. So these are the major deficiencies that we had identified so far in the plants. So nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, iron, manganese and zinc. These are the major deficiencies identified. Coming to the data sets, we could find 5 data sets where most of the plant images are present. The last 2, I think textual parameters are also present. But one thing to be noted is not images or textual parameters for all the plants are available in these data sets. It is limited to some 200 species of plants, I guess, but not all the plants. And one challenge that we found in these data sets is that the images and the parameters of the conventional methods of cultivation is what is available. When we come to hydroponics and aeroponics, those images or those textual parameters are missing even in these data sets. So, yes, so anything related to hydroponics and aeroponics, we are still in the verge of creation of data set. Deep learning methods that are working well in the conventional soil cultivation plants is found to not work ideally for the hydroponics and aeroponics plant. Universal CNN model, as I told you, one model which can be a benchmark for all the classification of plants, that is still yet to be found. So these are the research gaps. Deep networks are focused on biotic stresses, but not on abiotic stresses. The identification of the diseases, as my colleague told, identification of nutrient deficiencies is the first and foremost thing which is to be done, which can prevent the plants getting attacked by diseases. So that is the second one. Third one is the deep learning networks, which are supposed to be found for ideal classification. And then detection of stress and severity levels in the earlier stage. So these are the references. So from this, what we have found out is creation of data set for hydroponics and aeroponics plant, and then creation of a model to identify the nutrition deficiencies in these plants, because the nutrient deficiency symptoms in the hydroponic and aeroponic plant, the symptoms, if you see, it is not as same as what we do it in the traditional soil cultivation. So creation of data set and then a proper classification for the identification of nutrients is what we have planned. Thank you. Any questions? Yes, sir. Yes, sir. Yes. 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 Yes, sir. Yes. Yes. Okay. From the papers that I've gone through, I found a data set has been created for rice crop, which has been cultivated using hydroponics. But the images were collected using a drone, not with the satellite imagery. So there, the images were, the saplings were, immersed into solution, and then 10 different types of nutrition deficient solutions were taken, and then one solution with all complete nutrition was taken. So totally 11 classification. For rice, I was able to find 2 papers. under hydroponics environment. And the symptoms that it reflected, for example, a potassium deficient rice plant reflected a symptom of turning into light purple color, whereas the same thing when noticed with the conventional soil cultivation, it was not of purple color. So the symptoms also varies when when we do it with hydroponics and in the conventional method. So that is where, but I was not able to find out data set for hydroponics environment. So creation of data set is what we have initiated first, and then we are in the verge of doing the further identification of nutrients in hydroponic environment. So any other suggestion? Okay.
Thank you, ma'am.